the Lomonosov University, Konstantin Anokhin, to give a presentation, Brain and Cognitive Information from Biological to Artificial Intelligence. Please.
Yeah, I will repeat. I will start with the presentation on a project which is called Brain and Cognitive Information from Biological to Artificial Intelligence. Doesn't work, probably you can help me. Yeah, I will ask probably to place the slides. Yeah. If to speak about fundamental neuroscience, its main unresolved task is to understand the nature of cognitive transition. This is how cognitive systems develop from non-cognitive ones in the course of evolution. Importance of this question among many fundamental issues is also that it might lead to development of new generation of artificial intelligence, of artificial cognitive systems. Addressing these issues is the goal of a new project which started this year. It is funded by the program of the Presidium of Russian Academy of Sciences through the Ministry of Science of the Russian Foundation. The title of the project is Brain and Information from Natural to Artificial Intelligence. And its main idea is that cognitive systems differ from non-cognitive ones by their ability to generate, store, and use cognitive information. Therefore, the nature of cognitive information, that is encoding by the system elements within the system, inside the system, of interaction of the whole system with its relevant environment, is the main focus of the project. The project is directed by an outstanding mathematician, Professor Viktor Sadovniky, who is also the Vice Chancellor of the Moscow State University, and the project is hosted at Moscow State University and involves also two institutes of the Russian Academy of Sciences, Institute of Higher Nervous Activity and Neurophysiology, and Marshuk Institute of Numerical Mathematics. There are about 10 participating scientific groups in the project in the fields of molecular genetics, biochemistry, cellular biology, neurobiology and neurophysiology, neurophotonics, neuromorphic photonics, and artificial intelligence and deep neural networks. Our background in this project stems from the traditions of Russian physiology of higher nervous activity. The famous Pavlov's theory of conditioned reflexes. It was further developed in the 20th century post-Pavlov into the theory of functional systems. And today we work with the theory, which is an extension of functional systems theory, which is named neural hypernetwork theory. Its main idea is that cognition is maintained through the operations and the structure of neuronal hypernetworks, which bear the properties of carrying and processing cognitive information. The cellular basis of these mechanisms were well studied even in the Soviet neurophysiology with the single cell recording techniques in animals. There have been thousands of papers in this field of studies. However, the difficulty of studying such distributed functional systems which maintain intellectual behavior is that they are distributed over the vast areas of the brain, involving individual neurons into collective operations. And with the traditional tools of neurophysiology, it is very difficult to observe and to study such systems. However, we have developed in the course of the few recent decades a set of new tools to image these systems in the brain at the cellular level, to interrogate the issues of their formation and operation. The background for this is the discovery that during acquisition of new information, besides electrical activity guided by the large networks, in the neurons which are included into the new functional systems, the signals are generated which goes to genomic level to store new cognitive information. And with this discovery, it is possible to use the genes which are turned on by the activity in the brain to map functional systems which are established de novo for the first time with a new cognitive experience. And therefore, we developed a methodology which allows, besides electrical activity of neurons, to measure genomic activity, which can be called genomic action potentials, different from the traditional spikes of membrane action potentials. The idea is that gaps, as we call it, are cellular signals for encoding cognitive information. And therefore, by observing the activity of the signals at the molecular level in the whole brain, we can map the activity and localization and functions of new cognitive neuronal networks. To achieve this, we developed in 2003-2009 a set of new technologies to observe at the cellular level the activity of the whole brain. We developed special solutions which allows to make the brain transparent. So that here you can see, for example, the brain before optical clearing and after clearing. And also, thank you very much. We developed tools for microscopy of the whole body of the embryo, or for our cases, the whole brain of the animal, with the single cell resolution, which allows to map the activity of genes involved in the acquisition of new cognitive information at the cellular resolution within the whole brain. The next step is that we use the transgenic technologies, especially technology of trapping of activity of these genes, which involves the use of recombinase, the genetic recombinase, which allows, when the new information comes to the nervous system at the particular moment of subjective experience, to trigger recombination in those cells which are turned on to store new information. This recombination can lead to several results. First, it can introduce the fluorescent proteins, genes for fluorescent proteins. So we can identify the whole system ensemble of neurons which was involved in the new experience. Second, it is possible to introduce through recombination in these cells the photosensitive genes for photosensitive receptors and ionic channels, which allows, using light, to manipulate the parts of these large functional ensemble in the brain to see whether we can retrieve and complete the whole experience acquired by the system, whether the stimulation of particular parts are important for completion of the whole pattern, so-called pattern completion, and other issues of controlling this behavior. And fourth, we can observe introducing the calcium sensors or other sensors of neuronal activity. We can observe the activity of the brain in the cells which were involved in a particular cognitive experience. So we can label them and follow them through the life of the animal in other types of experience. So here you can see, can you try to put, yes, to start this video, please. Thank you. Yes, excellent, thank you. This is the three-dimensional reconstruction of the mouse brain with the optical clearing technology and light sheet microscopy that we have developed, and labeled with the genes for green fluorescent protein located in particular neurons. So at a higher resolution, we can see and study localization and activity of all the neurons which are distributed in this brain. This is ex vivo mapping. So this is done after a single experience of the animal and post-mortem. However, we have developed also the techniques based on the two-photon microscopy and mini microscopy, which allow to study the cognitive activity in intellectual tasks like this with the in the same neurons. It is better to probably switch off the light for a moment. In the Спасибо. So if you start, you can see these are the neurons which are labeled with the genetic sensor localized in specific cell types. And here under the microscope, this is the place for the objective of microscope, which is removed. We can see with the two-photon microscopy the cortical part of this large functional system firing when the animal is doing a particular cognitive task, solving a particular intellectual problem. So by using these tools, we ask in the project the following questions. What is the nature of cognitive information in biological networks? What is cognitive information mechanistically if to dissect it in the nervous system? Can we trace it? Can we define it and go to most elemental unit of cognitive information? So what is one bit of cognitive information in neuronal network? How it is encoded? How it is acquired and allocated to particular neurons? Why those but not other neurons in the vast ensembles of the cerebral cortex or other brain areas are triggering the acquisition of cognitive information in particular tasks? What are the rules for this? How this information is stored in individual neurons for a long time, and how it is retrieved from memory on demand during intellectual tasks. And using all the techniques and methodology which I described above, is it possible to measure quantitatively cognitive information in the experimental situations? Can we count the number of bits of cognitive information acquired by the nervous system in particular situations in different animals in different conditions? We'll believe that the answers to all these questions are feasible. And the second part of the project carried out by experts in artificial intelligence, mathematics, theoretical physics is to use the theory developed from these experiments to implement it for the development of artificial neuromorphic systems, which are based either on the deep neural networks, and the second direction which we are developing the elements for neuromorphic architecture based on photonic technologies, physical elements using neuromorphic photonics. Thank you.
Thank you, academician Anokin. Dear colleagues, I would like to pass and so asked to continue our forum, Dr. Otek. Yeah, please, Dr. Otek.
Thank you very much for Dr. Vladimir Cheonin. Yeah, I'm the chair, and I'm so pleased and also honored to be here in 4th Korea-Russian Science Day. And I'm a co-chair of this session and also a speaker. So next topic is my own presentation. Can I have first slide, please? Yeah, let me introduce one interesting single molecule instead of systemic neuroscience. So this is tentonin-3, which is a molecular sensor for mechanical stimuli. So let's think about mechanotransduction, which is a conversion of mechanical stimuli into biological science responses. And it's also very basic fundamental physiological processes such as touching, balancing, proprioception, hearing, and baroreceptor function. They all require a mechanotransduction. And also this mechanotransduction is mediated by mechanotransducer channels, such as, here's a couple of examples for mechanosensation. One is touching. So this is skin, and there are many organelles. It's in the sensory nerve. And these organelles contain mechanotransduction channels. And hearing also, we need a mechanotransduction channel for hair cell movement. Once hair cells are moving, then this channel is open and we can hear. And also blood pressure changing is sensed by mechanosensitive channel in baroreceptors. And also proprioception, we need to sense muscle length changes. So whenever we flex or extension, then muscle length is changing. Then mechanosensors must sense these changes of muscle length. These are all mechanosensation. There are, let's see, right. Okay, when you actually we culture sensory neurons such as DRG neuron, which is a somatosensory ganglion neuron, and you can see the soma of cultured DRG neuron. And once you patch on them and then you give mechanical stimulus, then there's a current, we call mechanosensitive current. There are 3 types of mechanosensitive current in DRG neuron. One is rapidly adapting, which is means that whenever you touch it or whenever you give a mechanical stimuli, then it's a rapid activation of currents, which is followed by very rapid inactivation. So it opens very rapidly and closes very rapidly. So we call rapidly adapting currents. And then the rightmost one is slowly adapting currents. So it opens rapidly, but it also closes very slowly compared to others. So inactivation time constant is around 330 milliseconds, which is compared very sharp contrast to rapidly adapting, which is only 3.7 milliseconds. So, and there are a couple of many genes, such as DEC-ENAC gene in C. elegans, and also Drosophila, there's a TRP channels. And in members, they call it a TMC channel, which is responsible for hearing. And MSCL channel is in bacteria. So there are many candidate channels, but these channels may not be a real mechanosensitive channel, because when overexpressed in heterologous system, it does not give some mechanosensitive current, except piezo family, which is discovered by Pataputian's group about 10 years ago. And this piezo is a very long and large membrane protein. It has more than 34 transmembrane domains. So when you overexpress and then you give mechanical stimuli, then there's a rapid activation and rapid inactivation, just like RA current in DRG neuron. So we know that RA current in DRG neuron is mediated by piezo 1 and 2. But then, what is the molecular identity for SA, mechanosensitive current in DRG? So we are trying to figure out, actually trying to find the molecules or genes that specifically mediate this SA mechanosensitive current in DRG. To do that, we have some specific cloning strategy. We actually use DNA chips, and then we differential hybridize of this DNA chip with the DRG neurons and many other cells. DRG neuron has SA mechanosensitive current, so it should contain genes specific for mediating that SA current. But other cells don't have any mechano responses. So we have very many candidates. Among these genes should have more than 4 transmembrane domain, because these are channels. And then we narrow down to about 40, 50 genes, and then we make each genes of siRNAs and then transfect it to DRG, testing for the loss of SA currents. Then we specifically, we are interested in TMEM150C, very strange names, it's code names. So we later, excuse me, renamed tentonin 3. In Greek, it's tension. So tentonin 3, we also call TTN3, is very small molecule. It's only has 290 amino acid and have a 6 transmembrane domain. This tentonin 3 has 2 more isoforms, so altogether 3 isoforms. We call tentonin 1, tentonin 2, and tentonin 3. And we found that this tentonin family are present only in vertebrates, not other lower animals. It's quite contrast to piezo family, which every species has piezo. So when we overexpress tentonin 3 in hex cell and then give mechanical stimuli, then we found that large mechanosensitive currents occur after transfection, which is very surprising because not many genes give this mechanosensitive current. This specific mechanosensitive current is just like SA current in DRG neuron. So, and then we check, we generate tentonin 3 knockout mouse. This knockout mouse shows 3, well, wild type knockout mouse, DRG neuron shows 3 different mechanosensitive currents, such as RA, IA, and SA. But in knockout mouse, only RA and IA currents are there, but the SA currents are completely wiped out. And you can see the proportion of cells in right side. So only the proportion of SA currents are reduced. So that means tentonin 3 mediate SA mechanosensitive currents in DRG neuron. And we found that tentonin 3 is cation channel, and also tentonin 3 currents are blocked by many mechanosensitive channel blockers, such as gadolinium, FM143, and GSMTX4. GSMTX4 is toxin from tarantula, it's spider. It's a well-known toxin for specifically inhibit mechanosensitive channel, and now we know that this toxin also inhibit tentonin 3 currents. So altogether, we can summarize that tentonin 3 is true mechanosensitive channel because it is activated by mechanical stimuli with a slow inactivity kinetics. So it's probably mediate SA mechanosensory current in DRG neuron. It is a cation channel blocked by many mechanosensory channel blockers. All right, let me introduce another story. This is about baroreceptor reflex. Probably you're well aware that what is baroreceptor reflex. Baroreceptor reflex is specifically to stabilize arterial blood pressure. So it's in carotid sinus and also aortic arch. So in aortic arch and carotid sinus, there's a specialized sensory nerve, which we call baroreceptor, which senses blood pressure changes. And we believe that 1023 is a mechanosensory channel and probably mediate this baroreceptor function. So when blood pressure goes up, then baroreceptor nerve activity goes up, which goes to transmitted to a cardiovascular center in brain, which actually reflexly inhibit sympathetic nerve and also excite vagus nerve. Thus, it reduces blood pressure as well as heart rate. So that compensates for the blood pressure increase. So this is all loop which controls blood pressure. And we know that 1023 is expressed in nodose ganglion, which goes to aorta. And we check all aorta projecting nodose ganglion neuron, and then we test for mechanosensitivity. The wild-type mouse nodose ganglion neuron has three, very interestingly, the three different mechanosensory currents, such as RA, IA, and SA. But 1023 knockout mouse, the nodose ganglion neuron shows only RA and IA, not much SA. So that means 1023 also mediate aorta projecting nodose ganglion function, especially SA current. Actually, this is published in JCI early this year. And then we inject actually AAV virus containing YFP. Actually, we generate 1023 Cree mouse first, and then we inject AAV virus that express YFP only in 1023 positive neuron. And then we realized that we can see very nicely about the nerve terminals that project to aortic arch. So actually, I cannot show you the movies, but well, you can see the fine fibers of this in the aortic arch, which express 1023. And then we make isolate aortic arch together with the nerve, which the nerve is called ADN, which is aortic depressor nerve that contains baroreceptor. And then we pressurize the aortic arch from 0 to 50 and 150 millimercury. As soon as we increase this intra-aortic pressure, then you can see the nerve activity goes up. But knockout mouse, it also shows some increase in nerve activity, but not as much as wild type. So there's a clear decrease of nerve activity to the aortic arch pressure. And then we recorded arterial pressure or blood pressure for 24 hours using a telemetric receiver. So you can see we measure 24 hours, but you can find that the knockout mouse has mean arterial pressure quite higher than mean arterial pressure of wild-type mouse. And also heart rate also goes up, which is exactly the same as the one that denervated those baroreceptor nerve. So once you remove the control system, then the arterial pressure goes up and also heart rate goes up. So that means that 1023 actually senses blood pressure changes in aortic arch. And we also inject phenylephrine to increase blood pressure. Then you can see the reflex decrease of heart rate. But knockout, you cannot see much. So in right panel and the lower bottom, then the baroreceptor sensitivity of wild type, the white one, is much bigger than knockout, the red one. So knockout has much reduced baroreceptor sensitivity. Then using a chemogenetic technique, we can express inhibitory as well as stimulatory response once you inject CNO, the agonist of this receptor. Then once you stimulate 1023 positive containing neuron, then you can see the green line that there's decrease in blood pressure as well as heart rate. But if you inhibit the red line and then you inject CNO, then there's an increase in blood pressure as well as heart rate, which is exactly as expected for baroreceptor function. And then we overexpress 1023 in 1023 knockout mouse to see if this rescue the impaired baroreceptor function in knockout mouse. So the bottom and left, 24-hour mean arterial pressure was high in knockout mouse, but once you overexpress 1023 in that knockout mouse, then you can see the rescue effect of blood pressure. In the middle, that's the heart rate. And rightmost, you can see the baroreceptor sensitivity. So the white one is wild type, has this much of sensitivity, but knockout goes down. But if you rescue one, if you overexpress 1023 in knockout mouse, then you can see the recovery or rescue of the sensitivity. So overall, we can see that 1023 is a mechanosensory channel or a molecular sensor for sensing pressure in aortic arch. Well, I thank to my students and also postdoc, Kyu Sang-hong, who first discovered 1023, and also Han-Jun Lu, who worked on mostly on baroreceptor function. Thank you very much.
Thank you very much, Dr. Matek. Thank you very much for your
You're welcome.
presentation. Dear colleagues, we will continue our work, and I would like to ask the correspondent member of Russian Academy of Sciences, Mr. Alexei Semyanov, to give a presentation, neuron-glial interaction as a basic for the brain function.
Good morning, colleagues, and good afternoon, probably, colleagues in Korea. And it's my pleasure to present at this meeting. So let's just see this when the slides will appear. So, okay. So this is the next talk, actually. Okay. Meanwhile, my name is Alexei Semyanov, and I'm working at Institute of Bioorganic Chemistry in Moscow. So this is my introduction slide, and I would like to start with general introduction. So we already heard something about neurons today, and since discovery of electrode-based methods, we know that the brain is an electric organ, and actually electrically excitable cells in the brain are neurons. And because of that, for a long time, people believe that neuronal network is the main cellular network in the brain. Neurons connected to each other by plastic synapses, and these synapses can change their strength, the network can be rewired, and therefore this is responsible for learning, memory, mind, consciousness, and everything, all functions of the brain. However, from physiological point of view, this sort of scheme of the brain function is a bit limited. Because just think if you take one cell type from the organ and try to explain the function of this organ, like take one cell type from heart or lungs and try to explain it. In fact, brain consists of many other cell types, which are jointly called glial cells. There's different subtypes of glial cells exist, and also blood vessels, lymphatic system, and space between cells filled with extracellular matrix. So all these components of the brain actually interacting to each other to make this sort of cell ensemble of the brain responsible for all the function of this organ. And today I will be talking about one type of non-neuronal cells, glial cells called astrocytes, and their interaction with neurons. Here you can see a schematic how the neurons and astrocytes interacting. This scheme shows the synapse, which consists of presynaptic part, postsynaptic part, and fine process of the astrocyte called leaflet. And this process of the astrocyte makes this synapse three-part type. So presynaptic, postsynaptic, and third partner is an astrocyte. And during synaptic transmission, a neurotransmitter targets not only postsynaptic receptors, also targeting receptors on astrocyte and triggering calcium response in the cells. And calcium activity in astrocyte will produce release of glial transmitter, which will affect release of neurotransmitter and will target also postsynaptic receptors, basically regulating transmission in the synapse and synaptic plasticity. On the level of entire astrocyte, here you see an astrocyte, calcium activity might occupy the larger territories, and this would modulate activity not in one single synapse, but might modulate activity in clusters of synapses. And these clusters might belong to one neuron or maybe belong to different neurons. These neurons might not even be synaptically connected, but they will be still modulated by the same calcium sort of event in astrocyte. And on the level of network interaction, astrocytic network, astrocytes also form a network in the brain, can interact with neuronal network. Activity of neurons will trigger calcium activity in astrocytic networks, and calcium activity in astrocytic network will affect neurons, vice versa. And therefore, this neuroglial network, that's how we sort of basically see the brain, these days actually become functional. And therefore, also, it's important to understand the mechanism of calcium activity, how calcium activity is actually generated and stored and maintained and integrated in astrocytes. We started our research a while ago. We started our research on astrocyte physiology and calcium activity in astrocyte more than 6 years ago. In 2014, we published the paper showing calcium pattern for calcium activity in cultured astrocytes. We were the first to identify individual calcium events. We analyzed frame by frame in time-lapse imaging, and we detected calcium events, as you can see here. These calcium events might be binarized, so we can measure duration of such events, then we can reconstruct these calcium events, and in 3 dimensions, X, Y, and time. And we can also get the maximal projection, so this would give us territory occupied by this calcium event size. And here you can see and appreciate several examples of calcium events taken from one astrocyte. Some events are very small, so this is again XY time representation. Some events are very small, some spreading over larger territories, and they also have very different duration, so that they have properties which can be measured. And we measured the duration, we measured maximum projection, and then we got distributions which are presented here in double logarithmic scale, and this signature of power law distributions. This is a very special type of distribution, requires a special introduction, which I cannot do with the short talk, but what I would like to show you here, so this is again power law distribution and the schematic of one astrocyte. You can see that astrocyte can generate events of very small size and larger size, which are much less frequent, but they are responsible for a significant amount of calcium activity. And neuronal activity not only triggering new calcium events, but also changing the properties of existing calcium events in astrocytes. And of course, it raises a question, how calcium activity is actually encoding some information in astrocytes. When they think of neuron, it's pretty much known. So neurons receive synaptic inputs. The synaptic inputs trigger dendritic spikes. Dendritic spikes propagate to the soma, and then in soma it's generated a pattern of action potentials, which is temporal coding. In astrocytes, it's slightly different. There is no integration. These events, they're not coming to one point to trigger larger events, so they form some kind of pattern. When I explain the difference, I always tell that there is like European languages, and based on the sequence of letters, this is how neurons code information, and languages based on Chinese characters or similar, when there is a pattern of activity which is carrying the information. And of course, it's very important to understand how this pattern of calcium activity in astrocytes is generated. To address this question, we performed some morphological study. This is a three-dimensional reconstruction of astrocytic processes, which is done with serial-section electron microscopy. And what we've done here, we reconstructed the form of astrocytic processes, and we also reconstructed calcium stores, which is responsible for amplification of calcium activity in astrocytes. And you can see here yellowish mitochondria and blue and red colored endoplasmic reticulum. The color indicates actually the size of endoplasmic reticulum within the astrocytic process. And we look at the distribution of this endoplasmic reticulum sizes, and we found that there are several peaks in the distribution corresponding to just normal endoplasmic reticulum, tangles of endoplasmic reticulum, and fine endoplasmic cisternae. So these tangles of endoplasmic reticulum are particularly important because they might amplify calcium events in astrocytes and represent the hotspots. And again, we looked where the inside of astrocyte is calcium hotspots and tangles of endoplasmic reticulum are located. And we look at the branching points, and we found indeed that these tangles located in branching points. So branching points is kind of gathering all calcium activity from their daughter processes and then amplifying it. So they're making hotspot activity. And to prove it, so we perform calcium imaging. This was already done in brain slices, which were the animals were sort of transfected with AAV virus with calcium sensor GCAM6 under promoter of astrocytic promoter. And then you can see this is a movie showing the video of all these calcium events which are already detected and presented and through the color for you. And this is a beautiful presentation which I really like. This is not one single event in XY time, but this is all activity of astrocyte presented in XY time. This is a population of activity of single astrocyte. And you can see there are many events of different sizes. And then you can sort of get their mean projection, and you can see where the most of calcium events actually generated. And in the picture in the corner, you can see that in the branching point of astrocytic processes, you can see very bright spots. These are hot spots, and these hot spots exactly correspond to tangles of endoplasmic reticulum. So astrocytic morphology and subcellular distribution of calcium stores actually defines calcium activity in astrocytes. And this is one single calcium event, how it starts and how it's propagated in one astrocytic branch. So you can see frame by frame. So it starts outside of the branch, then it propagates to the branch, and you see the branch is become much larger and brighter. And here is another representation. This is a representation showing rise and decay times of this calcium event. Rise times presented in red, decay times presented in blue, and intensity of the color showing the higher or lower rise rate or decay rate. And basically, also we can see that in the branching point, we have much faster rise and decay times. And why I'm showing this? Because this can be used to characterize astrocyte again, and this is the average projections for rise and decay times, and you can see that they also make hotspots. These hotspots generate and integrate local calcium activity. So, and they're responsible for this pattern generations. Oops. I pressed something wrong. Oh, now it's fine. Sorry. So all this data which I showed you were obtained either in cultures or in slices. And this is not the best preparation to link astrocyte to behavior. And of course, to link astrocyte to behavior, we need to do in vivo experiments. And we done them in awake mice. We wanted to see how astrocytes link to calcium, calcium activity in astrocytes linked to behavior. So we transfected them with AAV. Again, they're expressed in the cortex GCAM6 calcium sensor. This is recovery after surgery, and I'm not explaining you this beautiful device which Konstantin Anokhin already introduced. So this is the air floating platform where the animal can run and being hefted. Basically, in a awake animal, we can see calcium activity, how it's linked to behavior. And this is how it looks like in our hands. So on the left-hand side, you can see the tracking of the animal. The animal will be running. On the right-hand side, you can see basically calcium activity in astrocytes, not the neurons. So first, when the animal stands still, there is no calcium activity. When the animal starts running, we start to see increase in calcium activity in astrocytes. When it stops, the activity also stops. And then we can analyze all this. And again, this is a representation which you already seen before. So on the left side, you can see XY time representation of calcium activity, but not in single astrocyte now, but in astrocytic network. And you also see the column, which showing the episodes of running activity of the animal. And when the animal starts to run, astrocytes start to get activated, so that they light. And then when the animal stops, the activity decreases, and again. And we measure different parameters of this activity in astrocytes. If you look at the very bottom, so the black trace, it's a speed of activity, speed of running of animal. And then on the top, we can see how the amplitude of calcium events. Then next graph is the overall territory occupied by calcium events. Next graph is the sizes of individual calcium events. Next graph is the frequency or how many calcium events are generated. And this is actually very important and interesting slide, because what we discovered when the animal just started to run, we see overall increase in calcium activity in astrocytes, and sizes of calcium events in astrocytes increase. But then animal was still running, but astrocytes already stopped. The size of events decreased, but the frequency of events reliably followed the speed of running. So two parameters, they describe different properties of calcium activity. So two parameters describing different types of behavioral paradigms. So when the animals start running or probably making decision, the astrocytic calcium will become larger. But when it's already running, so the frequency of calcium events in astrocytes encodes the speed of running. So this is not neurons again, but astrocyte and directly linked to behavior. So these are my conclusions, which I already somehow went through during this presentation. So neuronal activity changes properties of calcium events in astrocytes. Astrocytes in the plasmic reticulum calcium stores forming the hotspots in astrocyte, and astrocytic calcium activity, different parameters of astrocytic calcium activity linked to different behavioral parameters, like decision to start running, for example, beginning of running of animal, and speed of running. These are encoded by different parameters of calcium activity in astrocytes. And this acknowledgement, this is, I thank the members of my current laboratory, and since it was a project which we ran for some quite long time, we have former members who also were involved in these studies, and our collaborators in the University of Nizhny Novgorod and University College London, and Helsinki University, and probably there could be a larger list, but I would like to thank particularly these people. Thanks for your attention.
Thank you.
Thank you very much.
Thank you, Dr. Smilyan.
Thank you, Dr. Chan. So, okay, Dr. Chehonin.
Thank you very much.
You're welcome.
Dear colleagues, let me introduce academician Mikhail Ugrumov with the presentation the fight against neurodegenerative diseases as a global challenge of the 21st century.
That's okay. Okay, chairs, ladies and gentlemen, it's a real pleasure to meet here, to be here and to contribute to meeting, particularly with Korean colleagues. Surprisingly, I regularly visit different Asian countries like China, Taiwan, Vietnam, Japan, but I've never visited Korea. Well, so that's really an opportunity to establish direct relations with our Korean colleagues. Well, so and this follows from the title of my presentation. It will concern the fight against neurodegenerative diseases as a global challenge of the 21st century. Please know that I will concern mainly Parkinson's disease, but the methodology and the paradigm for the fight is the same for all the neurodegenerative diseases like Alzheimer's disease, Huntington disease, and others. Well, and certainly that is not a journalistic expression, a global challenge or of the 21st century. This is a reality because the incidence of neurodegenerative diseases and the cost increase tremendously over recent years. And according to the prognosis of the World Health Organization, these diseases would bypass cardiovascular and oncological in the next 15 years. Well, so that's really a dramatic problem that is forgotten a little bit because of the coronavirus, actually. Yeah. Okay, good. Well, let's start with the examination of Parkinson's disease as an example of neurodegenerative diseases. There are so many details of the pathogenesis, of the diagnostics, and of the treatment, which are reported at any Congress, international Congress. But let's say what is definitely known about Parkinson's disease, actually. Well, this is a long-term progressing development without manifestation of motor symptoms. The emergence of the first motor symptoms many years after the onset of neurodegeneration, at a loss of most of the nigrostriatal dopaminergic neurons, which control the movement behavior, the motor behavior, and the depletion of compensatory reserves of the brain. And as a consequence, this is a very low, a limited efficiency of traditional substitutive therapy with the agonist of dopamine. And what is a prospect? It's not very difficult to understand that this is a development of preclinical diagnosis and preventive neuroprotective treatment. Well, and so this is a trend, world trend, at least for recent 30 years all over the world. Huh? But what have we achieved in these 30 years of hard job? Huh? More than 1,000 papers were published in best journals like Science, Nature, and others. Hundreds of $millions were invested, were spent, but the main result is still there is no technology for preclinical diagnosis of Parkinson's disease. What does it mean? Is something wrong in the paradigm for the development of the diagnosis and treatment? Well, and you should raise a question. Is it possible to diagnose Parkinson's disease at the preclinical stage nowadays? I say yes, with a positron emission tomography. By the evaluation of the functional condition of the dopaminergic neurons, which are responsible for motor behavior. Well, and but still, it is certain that this technique will never be used for the prophylactic examination of healthy population because of the high cost and the high sophistication of this technique. What shall we do? We should create, for the beginning, a small risk group. A small risk group according somewhat biomarkers. And then we could check the failure of the dopaminergic neurons with a PET scan only after creation risk group. And what is the current paradigm for the development of a clinical diagnosis of Parkinson's disease, which I say is wrong? Well, so what is the concept? What the people proceed in searching for peripheral biomarkers, easily available biomarkers, from the idea that Parkinson's disease, as other disease, neurodegenerative diseases, is a systemic disease. And therefore, a number of brain regions, the degradation of a number of brain regions precedes the degradation of the nigrostriatal system, which is responsible for the development of Parkinson's disease. And therefore, it's very logical to equate the preclinical markers, peripheral biomarkers, as non-motor or premotor symptoms and changes in body fluids. I mean, in the cerebrospinal fluid, in the blood, etc. But what is the current methodology? That people, at least for 30 years, they search for biomarkers of preclinical stage, but in patients at the clinical stage, as the premotor symptoms and there's changes in body fluids. And this is not correct. This is a drawback because certainly there is no overlap. There should not be overlap for 100% between the biomarkers at the clinical stage and at the preclinical stage. And that not surprisingly, that all biomarkers detected so far in patients are non-specific. And therefore, I think this situation was described ages ago by Confucius. It's very difficult to find a black cat. In our case, preclinical biomarkers in a dark room, in our case, in PD patients, in the clinical stage, especially if there is no cat, if there is no overlap between clinical and preclinical biomarkers. Well, so, and what should be done? Well, so we hypothesize that most of the blood markers at the preclinical stage differ from those at the clinical stage. And it follows from the pathogenesis of Parkinson's disease. And the former are less numerous, preclinical markers are less numerous and more specific than the clinical markers. And we checked this hypothesis and we proved it by evaluating, by searching for 15 markers in the blood as a change in the metabolism of catecholamines and amino acids. And only 23% of the markers which were found in patients at the clinical stage were the case for animals at the preclinical stage, because we checked it using a model of the preclinical stage of Parkinson's disease in mice. Well, so that was the first fashion to upgrade the methodology which is currently used. And the second, and then we implemented the second technology to upgrade the development of the early diagnosis of Parkinson's disease by searching for biomarkers in the blood of patients at the prodromal stage of PD. Well, so there is a certain manipulation. First, we should find out the risk group consisting of elderly people without motor symptoms, but with premotor symptoms like impaired sleep, olfaction, and peristalsis. And then in these patients, we should confirm that there is a degradation of the nigrostriatal system as it happens in patients with Parkinson's disease, in true patients. And then we found the markers in the blood. And so far we tested more than 50 markers in the blood, and we found at least 15 real markers of prodromal stage of Parkinson's disease. Well, so we in neurotoxins, in sphingolipids, and many others. Well, so but all these upgraded methodologies should be not specific for Parkinson's disease, because the Parkinson's disease is a systemic disease with the same elements sometimes as in Alzheimer's disease and other. And therefore the question is it possible to develop an alternative specific this time technology for preclinical diagnosis of Parkinson's disease? And I say yes. We turned to internal medicine, and we found that there is that in internal medicine, people, they use for ages, for decades, very efficiently a so-called provocative test to make a preclinical diagnosis of chronic diseases. And the provocative test or challenge test is any procedure in which a suspected pathophysiological abnormality is deliberately and irreversibly strengthened, causing a short-term characteristic reaction, symptoms of the disease. Well, so we decided first in neurology and psychiatry to apply the same provocative test to Parkinson's disease and the neurodegenerative diseases. And first we had to find out what is a specific key, what is a specific element of the pathogenesis of Parkinson's disease. And these are certainly nigrostriatal dopaminergic neurons. And therefore the requirements for this provocative test should be temporal enhance of the nigrostriatal system. And it should be a reversible deception. And they used, as a provocative agent, they used alpha-MTOP tyrosine. This is a reversible and non-metabolizable inhibitor of dopamine synthesis, of tyrosine hydroxylase and dopamine synthesis. And here we describe how we developed this technique. Well, so first we make a dose-dependence on the action of alpha-MTOP tyrosine to the expression of tyrosine hydroxylase. And then we have demonstrated that at a certain dose, it provokes the appearance, a short-term appearance of motor symptoms in animals at modeling preclinical Parkinson's disease. But there is nothing in normal animals or in control animals. And this action is reversible and for a short time. And for the physician, for medical doctors, it is particularly important whether this, any provocative agent, whether it is toxic for patients, can be toxic for patients. I say no. Alpha-MTOP tyrosine is not toxic at all to patients. And there are two types of arguments. First, this inhibitor is routinely used, applied to normal people to study the molecular mechanism of dopamine neurotransmission in the nigrostriatal system. On the other hand, the same inhibitor is used as a drug, as a medicine to treat patients with the chronic stimulation of catecholamine production, like malignant pheochromocytoma. Well, so and finally, what might be the conclusion? Well, so I repeat again that the highest priority in neurology and psychiatry actually is the development of clinical diagnosis and preventive treatment. Well, and whether this is a illusion or a reality, I think this is a reality. And there are two approaches which serve to upgrade the traditional approach in finding peripheral biomarkers. This is a search for blood biomarkers in untreated TB patients at the clinical stage with their validation in animal models, mostly of preclinical stage of the disease. And then the search on the right, the search of the blood markers in subjects at risk of TB at the prodromal stage, selected by premotor symptoms. But there is only one specific approach which has been proposed by our team. This is a provocative test using a reversible and non-metabolizable inhibitor of dopamine synthesis. Certainly, we should create, using all these tests, we should create a risk group, and we should validate the efficacy by using positron emission tomography. Well, so I think this is a topic of the nearest future. Thanks you very much for your time, and I'm very glad to be here and to meet my foreign colleagues. Thank you.
Thank you. Thank you very much for your spectacular presentation. Dear colleagues, we have completed I was at the first part of today's forum, today's
Right.
meeting, and we have a break time.
For your nice talk. And yeah, Dr. Chekhov, I'm sorry, Cheonin, it's your turn, please.
Dear colleagues, our next talk on a very similar topic, research and development in the area of brain-computer interface technologies by Professor Alexander Kaplan. Unfortunately, Professor Kaplan has to give at the very same time an offline talk at Moscow University on Lomonosov Prize. He apologized for this, but he pre-recorded his talk, and we will play back it now.
Good afternoon, dear colleagues. I'm very happy to join our meeting, our Russian-Korean meeting, especially because of 14 years ago I was invited in Chunbuk University as a visiting professor for 1 year. And in that time, it was very good, nice work for me because we collaborate between my lab in Chunbuk University and in biological faculty of Moscow State University. I'm working in the area of brain-computer interface last 15 years. And I want to say that this area is very wide. BCI technologies are sweeping the planet. Look at the publication rates from dozens of papers just 15 years ago up to 800 papers every year now. In Russia, more than 20 laboratories engaged in the development of BCI technologies. From Vladivostok, near South Korea, to Kaliningrad in the West. Only in Moscow we have 8 labs, maybe more, in the area of BCI technologies. Let me give you 2 examples. The first from my laboratory at the Faculty of Biology, Moscow State University. We are developing different neurointerface technologies for medicine. Here is some example of P300 technology, neuronal interface. We have developed this technology to over 95 accuracy. Here is our NeuroChat product for stroke patients who cannot speak. We have some cases even in the US. NeuroChat allows patients to type text, save them, send and receive mail, manage a small number of devices, and so on. Now, NeuroChat is on the market. And you see, about 500 NeuroChat kits are now in the hands of patients in hospitals and in the home. Another example is the work of my colleagues from Pirogov Medical Institute. They have developed BCI based on motor imagery technology to restore movements in patients with stroke or cerebral palsy. Their product is called ExoHand and is also on the market now. Of course, we have in our laboratories and in Russia as a whole many other examples of the development of BCI. But now I would like to talk about the problems and prospects of the BCI technologies. What are the main challenges of the BCI technology? First one, it's very low bit rate for transfer information from the brain to computer. Maximum 25 bits per minute. Normally, character selection is no more than 8 or 10 letters per minute. But if we used only 2 fingers, we can type about 100 letters per minute. Of course, the technology just for the patients after stroke. Next one. We need a lot of resources for vigilance and attention to concentrate on the letters on the screen or the motor images in the head. Usually after typing, for example, 50 characters, it needs a rest. It's very difficult to work in such kind of technology for the normal person. At the moment, we have no ways how to decode the thoughts, sentences, and even words by EEG analysis. Of course, it's possible to use invasive technology, but it's not impossible for the healthy person. But we are looking for the future. How to help to the healthy person. Next problem. In total, it's possible to decode no more than 4 or 6 mental images or intentions in the imaging technologies, and no more than 1 command per 20-30 seconds with good accuracy. It's a big problem. What about accuracy? If we are talking about the technology with the decoding of intentions, the accuracy is not sufficient. Only 15-25% above the chance level of a random selection 1 among 2, for example, left hand or right hand. So their rate is high, 25-35%. How to develop the training technology in the recovery movements with so many errors? Next one. Even if we detect the movement intentions, it's impossible to decode the speed, direction, goal of intended movement in non-invasive BCI. About invasive BCI we are already talked, but you see it needs to open the scalp. Next one. If we want to get good skills for controlling external actuators in BCI, it needs a lot of training sessions, about 50 or maybe 100. And it's only for the 3 or 4 control commands. So it's too long experience, it needs too long experience to get good skills. What about the prospects of BCI technology? I think the main prospect is the integration of BCI with elements of artificial intelligence to create a two-way communication channel between brain and AI. And second one, if you will take into consideration the well-known plasticity of the brain and artificial neuronal networks, it can be assumed that a specific brain machine language could appear in such a channel. And as a result, the first and the second together will give us the prospect of significantly expanding the resources of the human brain and its health. I'm sure together we can solve the problem and realize the prospects in the area of BCI technology. Thank you very much for your attention, and see you next time without COVID.
Thank you very much for your nice talk. Let's move on to other speakers. Let me introduce Professor Kim Sung-shin from Hanyang University. Yeah, just would you go ahead, your talk, please?
Okay, it's a great honor to present my talk in this Korean-Russian Science Day, sorry. And I'm also very pleased that many researchers are interested in motor learning and control. And so my topic today, would you show yeah, okay, thank you. So my topic is motor learning and memory with multiple timescales. Okay, so coincidentally, today, 12 years ago, today, December 2nd, the most famous patient in neuroscience history, patient HM, whose actual name is Haley Morlison, has passed away 12 years ago today. So that's why I decided to start my talk by introducing him. Actually, in 1953, he underwent a brain surgery extracting his bilateral hippocampi to cure his epilepsy. After the brain surgery, it is successful to control his seizure and epilepsy, but he suffered from the significant side effect, which is he could not form a new memory. So that means he didn't remember anything happened after the brain surgery. But interestingly, so again, this one shows that his brain without a hippocampi. But interestingly, he could perform and he could learn the motor skills. So for example, here the task is like drawing a star between two narrow lanes. He was able to do that. That means, as you can see here, his performance, the error he made was decreased across different days. That means he could acquire the motor skills. But that shows the famous experiment shows that there are at least two memory systems, different memory systems in human brain. Today I will talk about the motor memory and how motor memory is represented as we learn motor skills. Okay, this is the favorite quote by Michael Polanyi that we actually know more than we can tell. So what does it mean? So that means we have we have explicit knowledge that we can things about that we can touch and see and verbalize. But there's a more portion of our knowledge is the tacit knowledge that Michael Polanyi called this as a tacit knowledge, which is implicit knowledge that we cannot touch and see and verbalize. That one of the examples that Michael Polanyi gave us was the motor learning and memory. Okay, I think I emphasized enough the importance of the motor learning and memory. Then why that's why I'm very fascinated by studying motor learning and memory, because one of the reasons is our life is a continuum of motor learning. So for example, when you were a baby, you should learn to crawl on the floor, and then you learn to ride a cart and learn to stand up, and somehow later you learn to drive a car, right? Actually, by the way, this is my 4-year-old kid. Okay, so that's why our motor learning is very, very important in our life. It's full of our life is full of motor learning and memory. So I will present the two recent studies in my lab. The first one is motor memory with multiple timescales in motor adaptation, which is the one type of motor learning. In this functional MRI experiment, we actually suggest a computational model of an memory architecture to explain the motor learning and memory. This simple model consists of the several linear state space models, equations, and these models assume that there are several different memories with different time scales. For example, fast memory and slow memory. For example, fast memory runs very fast, but it decays. The state of the fast memory decays very quickly. But slow memory updates its state very slowly, but the forgetting is very slow as well. So this is much like a computer system. For example, fast memory is like cache memory, and slow memory is more like hard disk that that can store more long-term information. Okay, in this functional MRI experiments, we designed two conflicting motor learning tasks. The first one is the task is called the motor adaptation task. There is a certain rotation between actual hand movement and the cursor movement. So, for example, 40-degree counterclockwise rotation and 40-degree clockwise rotations between actual hand movement and the cursor movement. So, subjects actually learn these two conflicting motor tasks in the fMRI scanner. While subjects learn this task, we actually recorded their brain activity using functional MRI. Okay, this one shows the actual motor performance of the motor adaptation task for two conflicting motor tasks. You can see here red circles and blue curves represent two conflicting motor tasks. So, and we actually developed a computation model to explain human learning behaviors. And this model actually predict the motor performance with very high accuracy. And then the importance of the model is the power to predict the dynamic pattern of the temporal pattern of the brain activities with a different time scale. For example, like fast memory fluctuated very quickly and decay very quickly, as you can see here in reddish curve. And bluish curve, it shows the more slower time scales, which is gradually updates its state, which means like it represents the long-term memory in the brain. So using this computation model and the dynamic temporal activity patterns, we search the reason which will correlate the different dynamic pattern of the motor memory with the different time scales. What we found was pretty much consistent with the previous studies. For example, like prefrontal parietal regions, we found more fast memory, which is related to working memory. But in the intermediate time scale, we found the area in the interparietal area, and then interestingly, in the cerebellum, anterior part of the cerebellum is related to the slower memory, but the posterior part of the cerebellum is related to the little bit more relatively faster memory. So this video, could you play the video? So yeah, this video shows that the correlated reason were different depending on the different time scale. So again, prefrontal parietal is related with the faster memory, and this is shifted from the prefrontal parietal to the down to the anterior cerebellar area with a slower motor memory, slower time scale of motor memory. And we also employed a multivoxel pattern analysis, which is the one example of the algorithm, AI algorithms, to discriminate the pattern, three-dimensional brain activity patterns, just like Go pattern in the game of Go. So using the three-dimensional voxel activation pattern, we could discriminate the task, whether a patient subject learned the task 1 and versus task 2. And then interestingly, the anterior cerebellar area, the decoding accuracy of the pattern classification significantly increased across the different sessions. That means it represents the long-term memory for different tasks were stored in that area, and anterior cerebellar area. Okay, and then this is the very recent work published in 2 months ago in my lab. Because the previous task was very simple and takes like about 30 minutes to learn the entire task, but we actually made a very more complicated task. Could you play the video? And in this task, subject wear the data glove, which is a MR compatible. Then that means like they can perform this task. And the task, there is certain mapping between actual hand movement and the actual cursor movement on the screen. And this is predetermined mapping. Subject try to hit a target on the screen by manipulating their hand, So and then target jumps around the 4 different corners of the screen, and and then every 5 seconds, and then by trial and errors, subject somehow learn to hit a target and control the cursor by manipulating their hands. This is a very interesting task, and initially you can see here from the left figure. You can see initially the beginning, the trajectory is very messy, right? That means subject just figure out how to control the cursor and to hit a target, but because target is jumping around the different corners of the screen. And then, but as our practice goes, the subject figure out to control the cursor by manipulating their hand. The right figure shows the actual aspect ratio of the cursor trajectory is a little bit like straighter as a practice. And then this one, bottom figure shows the success rate to hit a target is increased. And by the way, this experiment was done multi-day. Actually, subjects visited our lab 7 times. So first day, subject performed this task in the scanner, and outside of scanner, they practiced this task for five days. And then later on, in the 7th day, subject did perform this task again in the functional MRI scanner. And we actually compare the early stage of learning and the late stage of learning after 5-day intensive practice in the outside of scanner. What we found was very interesting. In the caudate nucleus, which is part of the basal ganglia, we found gradual transition of reward modulation in the caudate nucleus. That means we actually use the time-varying reward rate as a regressor, and then we found the correlated area in the caudate nucleus. And this shows that initially the head region of the caudate nucleus was significantly correlated with the success rate, but later on, the correlated area is transient to the tail region. But this one is interestingly is only for the trend mapping. Actually, subject learn 2 different mappings, but they practice only 1 mapping in the behavior session. What we found was that this is learning specific, right? And then and another very interesting finding, we actually could predict individual subjects' performance in performing this task. What we found was like a cognitive loop consisting of dorsolateral prefrontal area and head region of the caudate nucleus. With a stronger interaction between these 2 areas in this loop, the subject perform less. The performance of subjects is lower when this cognitive is stronger. And the sensorimotor loop, stronger sensorimotor loop, the subject shows the higher performance for this task. This is very interesting, and this gives us a take-home message that if you think too much about, cognitively thinking about the motor task, then you would perform less. Your performance would be lower. So this makes sense, right? When you think about the motor task, you don't think too much when you perform the motor task, right? This is the take-home message of this research. And this is conclusion. We actually found functional MRI evidence of motor memory with multiple time tasks in 2 different studies. First study that I showed was a very simple motor adaptation task. Actually, motor adaptation task is the adapting motor behaviors to perturb the environment. In this experiment, we actually perturb the difference mapping between hand movements and actual cursor movements, like rotated counterclockwise and clockwise rotation. And this is the type of motor learning, which is called motor adaptation. In this very simple experiment, we found the transit, multiple motor memory, the evidence of motor memory with the different time scales. And we actually, subsequent study, we developed a de novo motor skill learning, in which subjects learn the motor task, right? Motor task from scratch, right? Actually learn to control a cursor on the screen to hit a target, right? In this task, we found, again, in the basal ganglia activity, specifically like caudate nucleus activity, is transit from the head region to the tail region, which is very interesting. Which is comparable with the previous non-human primate study. Okay, my future direction of the study, actually, as I showed, the brain implements the learning algorithm with a complementary memory system and with a different time scales. So my question was like, in how we can connect this finding to the AI. First question is, if you see this learning curve, then human learn the task very, very quickly. Right? My first question is, why is brain so efficient? And in terms of like with the very small amount of learning sample, how can human learn this task very quickly? And then how can human brain generalize the learning across multiple tasks? For example, two different conflicting tasks, right? So this is my key question. I will do the experiments with study to compare the human brain performance and human performance and then machine learning AI performance. Okay, thank you. Thank you, and this is my acknowledgement and all my previous supervisor and my students. And yeah, thank you, and it's a great pleasure to present my talk.
Thank you, Dr. Kim. Thank you very much. So, Dr. Anokin, your turn.
Thank you.
You are.
Our next talk from the Russian side is Professor Sergei Illarioshkin. He is from the Department of Brain Research of Research Center of Neurology, and the title is Relationship of Clinical and Fundamental Studies in the Field of Neurodegenerative Diseases.
Good afternoon, everybody. And today, Professor Blumov has already characterized neurodegenerative diseases as a great challenge to modern society. That's right, of course. And in my presentation, I'd like to stress the importance of the close link between clinical and fundamental studies in the field of neurodegenerative diseases, as might be exemplified by our Research Center of Neurology, in which we have simultaneously at the same time both clinical and basic settings, clinical and basic laboratories, which represent, of course, great advantage to study neurodegenerative diseases. And first of all, I'd like to thank all my colleagues working at the Research Center of Neurology and other institutions across Russia with whom we collaborate in different fields of neurodegenerative diseases. And today I'd like to focus on genetic studies, some example of our results, cell models of neurodegenerative disorders, and animal models which are closely related to each other. And to begin with, I'd like to say a few words about genetic studies of neurodegenerative diseases. In the past, our group was the first in our country who filled up the genetic map of Russia from the viewpoint of frequency of particular mutations or particular molecular forms of neurodegenerative diseases like Parkinson's disease, autosomal dominant, autosomal recessive ataxias, primary dystonias, different forms of degenerative dementia, motor neuron disease, and so on. And these data are important not only from the viewpoint of structure, of genetic structure of neurodegeneration diseases in Russia. And owing to these results, we were able to recruit a unique group of patients carrying mutations in different genes, but who still remains clinically non-affected. So patients being at the preclinical stage of neurodegenerative diseases. And this cohort of patients allowed us to monitor the dynamics of neurodegeneration years before the manifestation of clinical picture. And for instance, in Parkinson's disease, we recruit two groups of patients, clinical healthy patients. One of them are carrier of mutations in different Parkinsonian genes, at least 5 different genes or other risk factors, and a group of patients without mutations, just normal populations. And you may see here that carrier of mutations in different Parkinsonian genes are characterized by slow and slow increase in the number of well-known and non-specific biomarkers of neurodegeneration process in Parkinson's disease, like REM-sleep behavior disorder, hyposmia, constipation, fine motor abnormalities, and so on. While a group of non-carrier of mutations basically remains stable over years. Moreover, as you may see here, over a 7-year period, already 4 persons from the group of high risk carrier mutations turned out to be affected by Parkinson's disease, and clinical diagnosis became possible, while the group of low genetic risk still remain non-affected. So you may see on this group of patients, unique patients, we can follow the dynamics of neurodegenerative process over years. You can develop some biomarkers, check your hypothesis concerning the biomarkers and development of Parkinson's disease, and try some neuropredictive strategies. And these studies allow us to establish some global networks studying genetics of Parkinson's disease, like here, you may see here in this paper, and Moscow and Russia is here also indicated on this map. Why it is important now? Because today, several programs have been launched several programs on the treatment of particular molecular form of Parkinson's disease, carriers of particular mutations, by one or another new agent aimed at influence a particular protein affected in one or another genetic form of Parkinson's disease. So this is the first time in the history of studies of Parkinson's disease in the world where we are starting right now to personally treat patients carrying one or another mutation in at least two genes, LAR2 gene and GBA gene. Those molecular products are already in our hands. Of course, these trials, clinical trials, would be impossible without such a network because each particular genetic form of Parkinson's disease is extremely rare, and we could not collect, we could not recruit such a great amount of patients if we would not be able to establish that kind of network. It's why genetic studies are very important on the collaborative basis, and I hope our colleagues from South Korea will also help us to develop these studies further. And well, these results have been widely published in the literature. And now I'd like to go on to cell model of neurodegenerative diseases. And also genetics is also important to study cell models because in our experiment, we used genetic forms of Parkinson's disease or Huntington disease or spinal ataxia using so-called personalized cell models with the technology of induced pluripotent stem cells. As you know, you can take any freely easily available tissues, so tissues like skin fibroblast, and to convert them into induced pluripotent stem cells, and then to differentiate into virtually any kind of cells, including neurons. And in our studies, we converted cells from genetic mutations of Parkinson's disease into dopaminergic neurons and into GABAergic neurons from patients with Huntington disease. Let me remind you that GABAergic spiny neurons of the striatum are the main target of pathological process in Huntington disease. And also we were able to establish motor neuron cultures from patients affected with genetic forms of amyotrophic lateral sclerosis. Very briefly, a few results demonstrating potential of this technology. And let me remind you that Huntington disease, this is polyglutamine disease is caused by mutation expansion of CAG, polyglutamine coding repeats within the coding region of the gene. And in the past, the majority of researchers in the world preferred to use alleles with 60 to 100 repeats, most severe mutations, in order to easier get some pathological cell phenotype. But this is quite far from clinical studies because these extremely large alleles are extremely severe and almost never occur in practice, and those carriers of these mutations are virtually unviable. And our group was the first who were able to establish cell models with quite usual in clinical practice allele with 42 repeats. And you may see here some pathological autophagocyte formation, some abnormalities and mitophagy, and some indentation, nuclear indentations as typical clinical, not clinical, sorry, pathological finding, a characteristic of Huntington disease. So this model clearly recapitulates not only clinical, but just pathological finding well known in Huntington disease. Moreover, we were able to show some abnormalities in the store-operated calcium channels in these models, and also we showed that application on one of the candidate molecule can restore calcium channels, and this molecule now is maybe regarded as one of the candidates to treat patients or at least animal models at the first stage of Huntington's disease. And you may see here an example from Parkinson's studies. So we showed that carrier of mutations in Parkinson's disease at the cellular level, at the level of dopaminergic neurons, are characterized by disbalance of different apoptotic factors, by clear abnormalities in the growth, peptide growth factor like DNF and so on. We established some organoid 3D structures from carrying mutations in Parkinsonian genes and showed that these organoids are characterized by clearly abnormal neurophysiology. And these parameters might be used again to monitor effectivity, for instance, of one or another different drug, neuroprotectors, and so on. And you may see here transcriptome analysis of neurons obtained from iPSC with Parkinsonian mutations, different mutations. And in neurons, for instance, carrying PARC2 mutations, typical Parkinsonian mutations, we found increase in the expression of more than 60 mitochondrial genes as a reaction to an increased number of abnormal mitochondria that can be seen at the cell level. And in neurons carrying PARC2 mutation, metabolism is going predominantly via glycolysis, again shown by different molecular products. And in neurons carrying PARC2, LRRK2, and GBA mutations, three most common Parkinsonian genes, one can see abnormal expression of the TRIM family genes, which are very important in neuroimmune homeostasis and neuroimmune relationships in one of the earliest stage in the development of pathological process in Alzheimer's, Parkinson, and other neurodegenerative diseases. And very recently, this year, we obtained glial cell culture from patients with Parkinson's disease, sporadic and genetic forms of Parkinson's disease. And we were able to show that, you may see here, that medium conditioned by glial cells negatively affect on the length of the neurite on the dopaminergic culture obtained from these patients. And again, showing important role of glial cells in the molecular pathogenesis of Parkinson's disease. And again, it might be a good model to test the effectivity on one or another drugs. And so again, these studies have been published in several papers and several conclusions. iPSC-based studies mean help us to get knowledge about basic mechanisms of neurodegeneration. This means personalized approaches to the treatment, screening of potential new drugs, correction of genetic defect by CRISPR-Cas9 technology. We have also such an experience. It's a good experimental, good model to study experimental aspects of neurotransplantation and the iPSC banking, of course, for the future for neurotransplantation in humans and animal models. Let me show you some recent results in Alzheimer's disease, relationships between neurodegeneration and systemic metabolism. And so we used a model of Alzheimer's disease with streptozotocin injections into the lateral ventricle of the brain. And streptozotocin is a toxin which selectively damages not only neurons, but also pancreatic beta cells. And we obtained clear abnormal behavior, as seen here on passive avoidance reaction. It's changes in memory of animals, but also we could see some abnormal aggressivity, which recapitulates some psychotic symptoms characteristic of Alzheimer's disease. So you see, it's a really good model you see here. And surprisingly, one applying high caloric and low caloric diet in rats treated with streptozotocin, you can see that a large proportion of streptozotocin treated animals became extremely obese. Its obesity is quite similar to the obesity you can see on the classical model of Zucker rats, very expensive, by the way, and our rats were even more obese than those animals you may see here. So we decided to study with an endocrinological research center in Moscow, study the mechanism of such a phenomenon. And you may see here this Alzheimer model, streptozotocin Alzheimer model, is characterized by glucose tolerance test abnormalities. And you may see here on these curves, and they are characterized by clear pathology of the liver, of the pancreatic structures, and the abnormalities in secretion, as shown here by the classical marker of neuroendocrine secretory cells. And you may see here that this model is characterized by the changes in the hypothalamus, the walls of the third ventricle, so structure responsible for metabolism, homeostasis in the organism. And you may see here the accumulation of beta amyloid here, and as shown here, yeah, here again some abnormalities in the tyrosine hydroxylase and the arcuate nucleus. Again, changes in the some glial markers and so on. So this is substrate for metabolic changes we could see on this model. And some conclusions from this experiment. Streptozotocin-induced changes in the hypothalamus in rats with Alzheimer's pathology are responsible for systemic abnormalities of glucose metabolism. And combination of the brain streptozotocin damage and high caloric diet may induce severe obesity. This is a relevant model to study new neuroprotective agent in neurodegenerative diseases, at least in Alzheimer's disease. And the proposed model is very promising in Alzheimer's, maybe Parkinson, Huntington disease, multiple system atrophy, for testing such anti-diabetic drugs as agonists of glucagon like peptide, exenatide, metformin, and so on. Why? Because these drugs have been proposed recently as a potential neuroprotector for Alzheimer's or for Huntington disease because of some new properties discovered in this well-known molecule. Just an example, a few papers about rebranding of known anti-diabetic drugs in the treatment of Huntington, for instance, or Parkinson's disease. And I think our models, it's a good first step to try to establish new kind of treatment to test this molecule at the clinical and at the preclinical stage of neurodegeneration, both in animals, in humans, maybe at the cell level obtained from induced pluripotent stem cells. This is again a good illustration of close relations, of close links between clinical and experimental studies, and such a close relation greatly accelerate translation of results obtained in basic studies into clinical practice in neurological clinic. Thank you very much.
All right, let's move on to the last speaker of this session. So that came from KAIST. Are you there? Oh, is it? Oh, okay, Dr. Kim, yes, we are waiting for you. All right. Dr. Kim, Anjun Kim, okay, do you hear me?
Yes, I do hear.
Okay, just go ahead.
Let me share my file with you.
All right.
Okay, thank you very much for the invitation. It's my great honor to be able to be part of the Korea Russia Science Day. And so I study autism spectrum disorders with a focus on NMDA receptor dysfunctions. And so I've been studying synaptic molecules, in particular the molecules at excitatory synapses, and that form the synapses and make the synaptic function possible. So among many synaptic proteins, I've been focusing on these postsynaptic molecules. In particular, a group of postsynaptic protein called the synaptic scaffolding proteins. So these proteins are abundant and contains multiple domains for protein-protein interaction. So they interact with each other and form a platform-like structure where many receptors and adhesion molecules and signaling molecules are gathered to mediate synaptic transmission and plasticity. And in addition to these scaffolding proteins, I've been studying synaptic adhesion molecules, and they are present at the presynaptic and postsynaptic side and meet with each other in the synaptic cleft, and to mediate the synapse formation and also determine the synaptic specificity and synaptic strengths. Quite interestingly, recent clinical studies have indicated that many of these synaptic proteins are actually associated with diverse brain disorders in the neurology area, also neuropsychiatric area. So including, of course, autism spectrum disorder or ASD. So ASD is a type of a neurodevelopmental disorder. There are two core symptoms. One is a social deficit, another is a repetitive behavior. ASD comes with diverse comorbidities, including intellectual disability, hyperactivity, anxiety, and epilepsy. The worldwide prevalence is about 1.5%. And for some unknown reason, autism is 4 to 5 times more common in males than in females. So it is believed that autism is a highly genetic disorder. 80% here means that in the case of identical twins, they're likely to develop the same autism in 80% of the cases. And in terms of genetic etiology, this autism is highly heterogeneous. And in the Simons Foundation website, you can find almost 1,000 genes. But these genes are mainly from the Western samples. So the genes underlying autism in for instance, the East Asian populations are not really known. So recently we started the whole genome sequencing analysis for Korean autism patient. So we recruited trio samples, about 600 families. And so we sequenced 2,000 people and analyzed how mainly the de novo mutations in non-coding areas could be associated with autism. We found some interesting mechanisms. And this article is up in the bioRxiv website. So continuing on with the background, and the treatment for autism is really minimal. And these two antipsychotics are useful for irritation associated with autism, but not the core ASD symptoms. Because of that, many researchers around the world are very busy and trying to identify key ASD mechanisms. And of course, the pharmaceutical companies are very keen to develop the first ASD drugs. So in order to develop the drug, we need to know the mechanisms. And many people seem to agree that the imbalance between excitation and inhibition is likely underlying autism. However, excitation inhibition is such a vague term that can be applied to neuronal cells as well as synapses, excitatory versus and inhibitory. So my lab has been studying NMDA receptor dysfunction as a potential mechanism contributing to autism. Again, so we've been studying these synaptic molecules with focus on ASD. So the first clue for the NMDA receptor dysfunction hypothesis for autism came from this study showing a neuroligin-1 knockout mice show decreased NMDA receptor function and show ASD-like repetitive behavior. And when given NMDA receptor agonist, and this phenotype disappeared. Soon after our study, in collaboration with Dr. Bong-Kyung Kang at Seoul National University and Ming-Gu Lee at Yonsei University, found that the mice carrying SHANK2, the synaptic protein mutation, show autistic like a social deficit, which is responsive to NMDA receptor activation. And there have been some other studies, and including our recent study on mice lacking NGL2 synaptic adhesion molecule. So all these results suggest that decreased NMDA receptor function somehow contributed to autistic-like behavior in mice, and also likely in humans. That at the same time, too much NMDA receptor function seems to be causing trouble. So these mice models show increased NMDA receptor activity that are associated with autistic-like behavior. So these results collectively suggest that bidirectional deviation of NMDA receptor function can actually cause autistic-like behaviors. This notion is quite important because in patient cases, some patients may have a decreased NMDA receptor function, and others may have increased NMDA receptor function. So for those who have a decreased NMDA receptor function, we need to keep them NMDA receptor agonist rather than antagonist. So by characterizing which direction the NMDA receptor function is deviated, we can rationally decide the proper treatment scheme. And then what's the coverage of this NMDA receptor dysfunction hypothesis in general autism? And here you're looking at the list of medications that have been tested in many clinical trials for autism worldwide. And then this section, middle section, are the candidate medications for social impairment symptoms in autism. And you can see two NMDA receptor antagonists, one NMDA receptor agonist, comprising half of the whole 6 medications for social domain in autism. So it seems that NMDA receptor dysfunction is quite important. This review paper came out many years ago. What about today? So you know that if you visit this website, and we can see the clinical trials under certain disease names, and when I type autism, and there are 9 phase 3 clinical trials, and one of them was memantine. Again, it's NMDA receptor antagonist, and suggesting that NMDA receptor dysfunction likely play an important role in the pathogenesis of autism. However, NMDA receptor function actually changes very dynamically before birth, also after birth. And so we have to take a closer look at the changes in the NMDA receptor function during the development of autism across the timeline. So in rodents, we mainly measure electrophysiological deficits, in particular NMDA receptor deficits, using mouse at the age of around 1 month or before 1 month. And also it is well known that autism is characterized by clear diagnosis before the age of 3. So, but the human age of 3 years old roughly corresponds to 2 to 3 weeks in mice, if you agree. So that means we have to take a look at the earlier stages and some synaptic deficits in much younger mice. So as you know, the NMDA receptor function rapidly declines after birth up until 3rd week and maintained at adult levels. But in a mouse model of autism lacking SHANK2, we found very interesting NMDA receptor hyperactivity at 2 weeks and hypoactivity at 3rd week, and also hypoactivity in other stages. So what is going on? So we hypothesized that maybe abnormal hyperactivity at week 2 might be causing compensatory changes, and this compensatory mechanism is probably overshoot by the time the mouse reaches the age of 3 weeks, and this overshoot after the ending of the critical period is somehow persist into adulthood. So how do we prove this hypothesis? It was rather simple. Then we try to correct this NMDA receptor hyperactivity at 2 weeks by memantine treatment. And indeed, this short, rather acute treatment of memantine was able to correct the NMDA receptor hypofunction at juvenile stages and adult stages. And this had a very, and we didn't have to give the drug later on. So that means early pathophysiology is important, and early correction of NMDA receptor function has actually long-lasting effects. In other words, we don't have to give the drugs for a long time to the patients. So far, I've focused on the temporal axis over NMDA receptor dysfunction hypothesis for ASD. And I told you the early pathophysiology and correction is important. However, the brain is wide, and there are many different brain regions and cell types. And so, for instance, which cell types are more important, glutamate versus GABA? In the GABA cell types, there are several different subtypes of GABA, such as PV and subneuron, and also BFE neurons. So which neuron is more important? If an NMDA receptor dysfunction hypothesis is so important for autism, what about directly just deleting one of the NMDA receptor subunits and see if, whether this causes autistic-like phenotype in mice? So this GLUN2B subunit by NMDA receptor was particularly interesting because it has been associated with many different neuropsychiatric disorders, including autism and many developmental delays, ADHD, and so on. So we have generated knock-in mice carrying this point mutation found in patients, which are supposed to interfere with the normal just domain-to-domain interaction in the extracellular region of the NMDA receptor and cause the destabilization of the protein structure and eventually degradation of the protein. So indeed, we found NMDA receptor function was decreased, and GLUN2B dependent LTD, long-term depression, was impaired in these knock-in mice. So does this change in NMDA receptor function actually cause autistic-like behavior in these mice? And it seems to be the case because when we activate NMDA receptor function early on, and it seems to be rescuing the electrophysiological phenotype as well as the behavioral phenotype at later stages. Again, this was early pathophysiology and only acute treatment, and that has long-lasting effects. So to summarize my talk, I've been talking about the NMDA receptor dysfunction hypothesis for autism, and previously many studies focused on late NMDA receptor dysfunctions, but we've been studying lately the early NMDA receptor dysfunctions. And also we've been focusing on specific cell types, such as GABA and PB, some BFE neurons. We have already published some of these papers. And then direct ablation of one of the NMDA receptor subunits, NR2B in this case, seems to be important, in particular at the early stages of muscle development. But as you know, the GLUN2B subunits are expressed in the later stages. So nowadays we are focusing on later stage pathophysiology as well. As I told you, there are so many autism-related genes, 1,000, because there are so many. The Simons Foundation and many scientific advisory board people just decided to categorize these 1,000 genes into, shown here, 6 different groups. So the genes are falling into this S group, or standing for the symptomatic group, meaning that the defects in a particular single gene can cause not only autism but other neurological and neuropsychiatric disorders. But this group belonging to the category 1, there are only 25 genes, and these gene defects mainly cause autism, but not other neuropsychiatric disorders. So this is actually a list of the 25 genes and their full names and their functions. Turns out that some of them are synaptic proteins, but many of them have just a non-synaptic function, such as chromatin remodeling or signaling regulation and transcription regulations. So now my laboratory is testing knockout animals or knock-in animals, just carrying some of these mutations, and try to test whether the NMDA receptor dysfunctions are also found in these mouse models. So this is ongoing and painstaking approach, but I hope I could be able to tell you whether or not NMDA receptor dysfunction is quite prevalent in animal models of autism, hopefully in the near future. Yeah, these are the list of the people who have been collaborating with us. Thank you very much for your attention.
Thank you very much, Dr. Kim. All right, this is the end of our session, and now we have to have a question and answer session. Dr. Anokin, are you there?
Yes, I'm here. I am ready to co-chair the discussion.
Right.
So our talks are open for questions and discussion, please.
Okay, let me ask Dr. Anjun Kim about NMDA receptor dysfunction.
Okay.
And then you told us that NMDA receptor dysfunction is related to GABA-erging neurons, but
That's right.
I cannot relate those two words because NMDA receptor should be excitatory, and how can it be related to GABA-erging neuron? Would you tell us some more detail about it?
Okay, I'm very sorry. My explanation was not enough. So NMDA receptors are present on dendrites of many different types of neurons, including excitatory, inhibitory, dopaminergic neuron, adrenergic neuron, etc., likely in the dorsal root ganglion neurons as well. So, so it is quite interesting to think about the defects of NMDA receptor on GABA neuron and also on excitatory neurons. Actually, the outcome would be different and actually opposite. So, yeah, we are trying to, it seems that the GABA neuron seems to be more important than excitatory neuron, at least for the few synaptic proteins that we've been testing. So we'll have to see. But the general consensus nowadays is that the GABA neuron seems to be more regulatory than excitatory neurons. So likely to play more important role in the pathophysiology and likely to function as an important target cell types for pharmacological or other types of intervention.
So what they are doing, what NMDA receptor in GABAergic neuron doing?
Okay. Well, their composition and the property seems to be slightly different. Okay. So the GABAergic neuronal dendrites have both excitatory and inhibitory synapses.
That's very interesting.
And their balance somehow determines the output of GABAergic neuron, the firing of the GABAergic neurotransmitter release onto pyramidal neurons.
So that means the presynaptic buton, they release GABA as well as glutamate. Is that what you're saying?
Actually, GABA neuron release only glutamate, but this is what's happening at the nerve terminal of GABA neuron. But in the dendritic area of GABA neuron, there are excitatory and inhibitory synapses.
I see. Yeah.
And NMDA receptor is an important
component. I see. Now I got it. Yeah, okay.
Sorry for,
May I also have an accompanying question to Anjun Kim?
Okay.
What is your view on the specificity of NMDA receptor dysfunction towards ASD? The question is, if there is a dysfunction of NMDA receptors during development in SHANK2 knockout mice, for example, or in other conditions, why you see the specific symptoms of ASD but not mental retardations, schizophrenia symptoms, etc.? And it is related also to the issue of treatment then.
So you're very correct, and a single gene cannot be associated with a single disease. As I already showed in the slide, and green-2B, for instance, encoding gluN2B subunit and the receptors, and have been associated with half a dozen brain disorders. So I doubt that a single gene is only specific for instance, NMDA receptor is only specific for autism. As you probably well aware of, NMDA receptor dysfunction has been one of the key mechanisms for schizophrenia and also intellectual disability, as you mentioned. So, however, NMDA receptors are not homogeneous in terms of their composition and function and sensitivity to neurotransmitter in different brain regions, different cell types. So when, for instance, GLUN2B subunits mutations mainly cause autism, whereas the GLUN2A subunit mutations mainly cause schizophrenia. So there seem to be some subtle differences that determines the specificity towards particular diseases, and also onset of diseases and the trajectory of the diseases and so on.
But in human conditions, we do not have the situation of comorbidity of ASD and schizophrenia. So the question is to which degree the experimental data on NMDA receptor dysfunctions are relevant to the clinical situation.
Yeah, you're very correct, and hard to tell. And actually, social impairment, for instance, are found in mouse modeling schizophrenia, ADHD, and autism. So behavior is quite a mess, I have to say. In terms of cell type specificity and neural circuits, and there are probably some shared circuits and differential circuits. And so your case of schizophrenia, intellectual disability, likely to share a certain neural circuits that applies to both diseases. But because we don't fully understand the neural circuits associated with schizophrenia, intellectual disability, the autism, it is at the moment quite a challenge to figure out, to understand the circuit specificity and how differential neural circuits differentially contribute to different extents.
Dr. Kim, thank you very much. We have to move on to other
questions.
Sorry, yeah. No problem. Thank you very much.
Thank you. From our side, Professor Ugrumov had a question.
Okay, thank you very much. So this is a general discussion. I should say that I was highly impressed by all the presentations. That was really top level. But I have a feeling that I'm the less educated guy in this audience, and therefore I have questions to all the speakers. Well, so and the first question is to Professor Anokhin. Well, thank you very much for your exposition of an excellent project on cognitive functions. Well, but my question is just very general. What is the originality and competitiveness of your project when comparing with similar projects in other countries? Brain Initiative in the United States, only the European Union and Chinese, etc. Very briefly. Well, so that is the first, but not the last. I will put all my questions, okay? Well, so then the second presentation concerned the mechanoreception and the very fine molecular mechanism of mechanoreception. That's very, very important. But my question is whether the impairment of mechanoreceptors is somehow related to neuropathology. If this is so, that might be a very interesting tool to develop or to improve the diagnostic techniques and to use this phenomenon as a test for testing of the efficacy of the treatment of patients in this neuropathology. That's very important. Well, so then there is a number of presentations which dealt with the interface brain-computer. That's very important. And some of them dealt with the or the use of the invasive interface brain-computer, mostly in monkeys. But for the improvement of the learning, memory, motor behavior, etc., etc. Well, but just to remind you that as far as I know, the father or the pioneer of this technique, that was the French neurosurgeon Benabit, who developed, in fact, this is a precursor of this system for the treatment of Parkinson patients with so-called high-frequency stimulation. That was in '87, okay? Since this time, this technique, it was not called as, at that time as interface brain-computer, but that was, in fact, that was the same. Well, but later, this technique is widely used actually for the treatment of depression, of epilepsy, etc., etc. But there are always the same complication. This is an irradiation of the action and side effects. And therefore, my question is to these people who reported this data, are there complications or side effects if you improve, for instance, if you try to improve the memory, for instance, by stimulating hippocampus or cortex? That is my question. Well, so then I have a question to Dr. Ilya, Professor Ilyoshkin, but very excited, very similar to my presentation in the idea of neurodegenerative diseases. Well, so in two presentations by Simyanov and by Ilya, it was focused on the important role of glial cells in the functioning of the brain and also in pathology like Parkinson's disease. Well, so my question is, do we have some arguments in favor of the primarily pathology of glial cells in neurodegenerative diseases? And if we have, please note them, okay? And then, and also I have another question, because Professor Ilya touched a very important problem of genetically predetermined Parkinson's disease, and particularly which modify the metabolism of so-called alpha-synuclein, which is very important in non-function, but it becomes toxic in Parkinson's disease because of the aggregation. Well, so we say that this is a general neurotoxic, but with a specific action to nigrostriatal dopaminergic neurons. Please tell me, what might be the mechanism of the specificity of this action on dopaminergic neurons of the nigrostriatal system? Nobody has an idea. Probably we have an original one. When, and the last presentation about autism. Also, I was deeply touched. That's very, very, very important problem. But it was said that that is mostly genetic or something like this for 80% disease. Well, so my question is whether we can differentiate autism like Parkinson's disease or like Alzheimer's disease for monogenic form and for a polygenic form. If we can differentiate it, could you say me what might be the epigenetic factors which trigger the development of autism in embryogenesis, I think, okay? And also, I know that in addition to this NMDA receptor concept, there was a very popular concept on the deficiency or the impairment of serotoninergic system. Could you tell me what is the situation actually with this issue? Thank you so much for my probably not, not very professional questions.
Thank you very much for your questions. Before I forget your questions, I have to answer to a few questions. The first two questions. Let me introduce our history of grant for neuroscience research in Korea. I think 2 decades ago, we started out small grants for neuroscientists only. And it was not a big one, but so 10 years ago, now it's second term, and they increased the grant a little bit more, but now it's a third term. Now they increase a huge amount of money. So let's see, how much does it go? Well, actually, $60 million a year for neuroscience only. So I think, yeah, over $60 million a year for neuroscience research these days. So it started out with that, and that does not include the research money for curing or studying Alzheimer's disease, which is another big grant. So there are fair much of money for neuroscience these days. So I think we all happy about that. And second question, to your second question, you said if a molecular sensor for mechanosensation is dysfunction in some patient, what kind of disease is related to the mechanochannel dysfunction? Actually, one paper actually came out. So that's a piezo-2 dysfunction in certain patient. The patient who has the mutation in piezo-2 genes, then they have a hard time to emptying their bladder. So they cannot control urination. So they have very many symptoms of, they call it lower urinary tract syndromes. So they pee quite often, and they cannot stand for urination. So and then yeah, very similar syndrome. So again, but there are many other diseases related to mechanosensation, because those sensory neurons have many different genes. So they are all like tentonin 3 and piezo 1. They are sort of, it's redundant. So one mutation of one gene doesn't mean that doesn't harm too much. That's what I'm thinking. So we cannot single out one mutation of one gene does very much harm to human functions. Okay, this is my answer. Thank you.
We can proceed with the list of Professor Agrumov. Thank you very much for giving the chance to emphasize the uniqueness of our project. It was on the slides, but I will summarize it in a different way. There are two fields at the moment. One is classical neurophysiology, with the electrophysiology of recording of neural activity. And the second is molecular neuroscience. With the first domain, the most striking phenomenon is cognitive specialization of neurons in the animal and human brain. They are called in a different way: grandmother neurons, concept neurons, smart neurons. The idea is that the individual cells are shown to be in different animals to encode knowledge. They show the response to different concepts. And this is learning dependent. The other area is molecular neuroscience, where the studies of synaptic plasticity, gene expression dependent learning has been for 30 years studying what are the molecular machinery of long-term memory plasticity. But none of the studies so far have attempted to bring together these two issues and to ask what is the molecular mechanism behind acquisition of knowledge in the individual neurons, how these neurons in molecular terms specialize towards a particular knowledge of the whole animal or the whole human being organism. The reason is there were no techniques to record individually in the neurons electrophysiology and genomic activity during the course of learning. And our project, for the first time, there has been no such studies. Therefore, I emphasize so much the techniques, is attempting and going to solve this project. And we have the preliminary results of how to do this. Professor Illarioshkin, probably from our side.
Mikhail Vladimirovich, thank you very much for your questions. Well, let me begin with the glial cells and the role of glial cells in the neurodegenerative. Of course, we have several examples in which glial cell pathology is primary to the development of neurodegeneration. For instance, multiple system atrophy. This is primary gliopathy because alpha-synuclein has been accumulated not in neuron primarily, in glial cells. It's well-known feature. So one of the quite frequent neurodegenerative disease, multiple system atrophy, is glial cell synucleinopathy, as compared to Parkinson's disease, which is neuronal synucleinopathy. Moreover, amyotrophic lateral sclerosis. So there are some very early marker of glial cell pathology and damage, which are now used in clinical practice as very early markers of neuronal dysfunction and or dysfunction of central nervous system in the amyotrophic lateral sclerosis. There are several molecules secreted by glial cells which it was shown in the experimental conditions might somehow trigger pathological process of like abnormality of motor neurons on cell culture on animal models. And moreover, as you know, there is some so-called lymphatic system, so specific lymphatic system of the brain, which is very important for the early pathogenesis of Alzheimer's disease, for instance. This is primary role of glial cells in detoxicating the brain, especially during the night sleep. That's why night sleep is a very important factor protecting from neurodegeneration of Alzheimer's type. This is role of glial cells in, in re-establishing some kind of detoxification is very important. Concerning alpha-synuclein, I don't think that nigrostriatal system and nigral neurons are especially vulnerable to synuclein itself. They are especially vulnerable to neurodegeneration, but the exact role of alpha-synuclein is not known until now. It somehow mediates neurodegeneration via the very, very complicated, network of events, but nobody knows exactly. And concerning the amount of alpha-synuclein, amount of neurons died, for instance, in Parkinson's disease, at the very early stage, and the number of neurons within the locus coeruleus, it's not dopaminergic, noradrenergic structure, equally as low as in Nigra at the very beginning of the disease. But we take notice on the sleep disorder, but we always notice motor disorder. This depends on the topic of your attention. But there are many other systems in the even at the very beginning of Parkinson's disease, apart from Nigra cell system, which already might be affected. It's very high level, but so you may see it's not so only just specificity for Nigra system. I don't think it's quite. We should be focused on alpha-synuclein, just some mediator, some well-known indicator, and very useful molecule to model some process within neural networks, but no more.
Few comments from Dr. Chang.
Yeah. For the BMI application, I think that still there are no application circumventing, overcoming the Parkinson's disease. But I think that there are several frontiers, including the sensory feedback application and some cognitive BMI approach, such as speech BCI, started from the UCSF. So, in Korean government launched cognitive BCI, particularly focusing on speech BCI. So I think that we are probably expanding those applications. But as you mentioned that there are many complications stemming from the use of the invasive electrode. So I think that with the nanotechnology, probably we could overcome those shortcomings of the electrode-related complications. So I think that the future of the BCI application is, I think that the expanding right now, not only in Korea, but also in the worldwide. So I think that Russia could contribute a lot more to the BCI research. Yeah.
So I have one question. Actually, few years ago, actually, it was published in, last year or this year, New England Journal of Medicine, that in Harvard Hospital, doctors they inject neural stem cell, which is derived from iPS of patients, fibroblast, and they inject it to a brain of the Parkinson's disease patient, and then they report that it is very helpful to the patient. So my question is that any other cases of neural stem cell injection to PD patients in Russia?
May I answer this question?
Okay. Well, thank you very much for this excellent topic that is raised in your question. This is a cell technology in the treatment primarily of Parkinson's disease. Well, so it started this this development. It started in the early 90s by Professor Björklund and by Swedish and by the European program, which was called NICTAR. And I was a participant of this program. And the idea was to implant human dopaminergic neurons of human embryos to the striatum of patients. Well, so Björklund has received an excellent therapeutic effects in experimental models in rodents, but we didn't have good results in patients in European countries. And that was the case for the United States over this period also in the 90s, 2000. But since this time, at least 4 generations of cell technologies were applied with the same aim, like genetically engineered cells, stem cells, and many others. Well, with the same results, excellent effect in experimental models in rodents, and almost no effect in patients. Well, so therefore, I left this area at the beginning of 2000 because I came to the conclusion that there is a sort of taboo in the implantation of neurons to the human brain. I think that because the original idea of Björklund was to implant a sort of pump of dopamine. It was not important what kind of pump. He selected embryonic neurons, but he could select any other thing, okay? But the brain functioning in rodents and in human beings certainly is quite different. And I have a feeling that in rodents, there is a preferable so-called volume neurotransmission. It doesn't mean to what region do you locally inject dopamine. Well, because the action is diffusive. But in human being, there is a preferable synaptic action, very local point action within the synapses. And in order to improve the situation in patients, we should reconstruct the microsynaptic organization. It means that we should develop 10 or 15,000 axonal efferents to any neuron in the striatum, and that is important. Therefore, I think that there is for the moment, there is a biological restriction for the use of stem cells or any other cells for the replacement of dopaminergic neurons. There is another side of the medal that we could use them as a source of growth factors, which stimulate the reparation, okay? But this is another point, completely another point. Thank you for your attention.
Well, thank you very much for your nice explanation. Thank you.
So I have a few questions about treating Parkinson's disease, and I'm pretty interested in transcranial magnetic stimulation. It's to treat, mostly known for its treatment effects of depression, right? So I wonder if is there any clinical approach of treating Parkinson's disease using transcranial magnetic stimulation? This is the question one. And my second question was to Dr. Konstantin Anokins. And is there any collaborate? I was pretty impressed by the DeepMind developing more algorithms like reinforcement learning algorithm, like beating human performance in gaming and game and others, right? And AlphaGo, for example. And is there any collaboration between computer science and like neuroscientists, computer scientists and neuroscience to develop a more human-like artificial intelligence systems? And this is my second question.
Okay, I'd like to. provide some answer to questions. So our Research Center of Neurology have some experience of transcranial magnetic stimulation in the case of primary depression, not in Parkinson's disease, but in patients with primary pharmacoresistant depression. This is our collaboration with the Research Center for Mental Health. And we have some very interesting results published already with the primary transcranial magnetic stimulation of the dorsolateral prefrontal cortex. This is ongoing studies that should be continued, but it's very interesting, very promising, because this, again, I would like to repeat, this was resistant depression, very difficult to manage patients. And well, not in Parkinson's disease, because this well-known experience of clinicians managing patients with Parkinson's disease show that depression in Parkinson's disease can be easily much more easily corrected by conventional drugs, which are in our disposal.
My turn. Professor Kim, I actually had a question also to you, but I will use my time to answer your question first, and then say probably a few words, because we are coming to the end of the discussion, to our Korean colleagues about the Russian Brain Project, the program which is under development and is chaired by the president of the Russian Academy of Sciences and vice president of Academy of Sciences, Vladimir Chikonin. He left, but I will say a few words. First, about your question. You are absolutely right. The main trend in the next generation of artificial intelligence, on which there is a focus in our country, is human-level artificial intelligence of general type. Probably you know that we have a federal research program on artificial intelligence in our country. And within this program, there is a chapter on the development of new techniques in the artificial intelligence. Within this framework, the main focus is artificial general intelligence. That is human-like artificial intelligence. The program is coordinated and was shaped by the largest bank in Russia, Sberbank, and it has established now the Institute for Artificial Intelligence, and it will focus on the issue of artificial general intelligence as a future development. This is first. Second, at the Moscow State University, where our project is located, this September, a new interdisciplinary school between different faculties was formed. It is called Brain, Cognitive Systems, and Artificial Intelligence. And the scope is that to bring the computer science departments, there are two faculties of mechanics and mathematics and computer science, with the life sciences around the research on the general artificial intelligence. And there are also two institutes formed. One I represent is the Institute for Advanced Brain Studies, and the second is the Institute for Artificial Intelligence Systems. And again, there is a cooperation. So it is exactly in the direction of what you have asked. The development is taking part. And to complete this, I would say that this is one of three directions of the Russian Brain Project, which is under the development, and it is in the Russian government, to be funded probably starting from the year 2022. It was prepared by a group of experts in the Russian Academy of Sciences, and we are part of this group and submitted to the president of the country and to the government, approved by the president and submitted to the government. You know that each national research project in neuroscience has its own focus. For example, in Korea, there is a strong focus on neurotechnologies. In United States, it's technologies for the study of the brain. European Brain Project, Human Brain Project has its own focus. Our focus in the program in Russia is first on three priority areas, three fields: fundamental brain research, clinical brain research, and new neurotechnologies. And each of it has its own specificity. In the fundamental studies, we have two areas of priority. One is molecular mechanisms and neuronal plasticity. So synaptic plasticity, gene-dependent plasticity, cellular processes. And the second is higher brain functions, including memory, cognition, and consciousness. In the area of clinically oriented brain research, there is a strong emphasis related to fundamental issues of neuroplasticity, learning, memory. It is the issue of neurodegenerative diseases with a specific focus on the early diagnosis and therefore prevention of the development of these diseases. There are also other areas in the child mental disorders, autism spectrum disorders, etc. But this is the main emphasis. And the third area I already described is neurotechnologies with the focus on brain-computer interfaces and neuromorphic artificial intelligence. So perhaps if we will move forward in our collaboration and cooperation between two countries and have some joint program of collaborative research, we can discuss the topics of the joint grants in considering these emphasis and priorities in the Russian Brain Project. Thank you.
Thank you very much. I think, Dr. Anokin, this is about time to wrap up our session. So would you give us your final comments or words to us?
Probably you can complete our session because I already made it.
I see, all right. So, well, colleagues, thank you very much, all speakers and chair. This is a great honor to be here. It's pity that we cannot sit together at the same place because of COVID-19 situation. I'm sure we're gonna meet together sometime soon. Then we discuss more about neuroscience and also questions and recent results together. But, well, thank you very much. So again, I think we need to close this session. Thank you. Bye-bye.
Thank you, and hope to meet all of you in person.
Great. Thank you.
Thank you.
Thank you very much.
Goodbye.